Welcome to my YouTube channel K Education. Please share, subscribe and like this video. Also press the bell icon due to which you will get notification of my new video. Welcome to this session. In this session we will study the biology first lesson, first part, heredity and evolution, 10th class, Maharashtra board, English medium. So in this lesson we will go through some points. Those points are heredity and hereditary changes, transcription, translation, translocation, evolution, evidences of evolution, Darwin's theory of natural selection, Lamarckism, speciation and human evolution. But today, due to only first part only I will tell you, so I will tell this first two points, that is heredity and hereditary changes, and transcription, translation and translocation. See, heredity and hereditary changes. First of all, what is meant by the word heredity? If you read the first two lines, you will get what is heredity meant by. You know heredity. See here, from heredity. Heredity is a transfer of biological characters from one generation to another, by via genes. This is nothing but heredity. Okay. Now, some scientists who made studies in a field of heredity. First of all, name is taken of George John Mendel. In 9th standard, we have studied about him. He was a pioneer. Of the modern genetics. It took a long time to understand the conclusion of his research about heredity. Next was Hugo Davis. Understood, or he studied the sudden changes in mutational theory. So he studied this mutational theory. Meanwhile, see, Walter Sutton is the third one. Walter Sutton observed the paired chromosome in cells of grasshopper. Until then, it was not known to anybody. Research started in direction of finding the nature of the genetic material when it was proved that genes are carried via chromosomes. See, this is the most important. Genes are carried via chromosomes here. When this thing was known to everyone, that time the study was started in a direction of what is in this word heredity and what is it about. Then a triad of scientists, Oswald Avery, Michael McCarthy and Colin McLeod, proved that except viruses, all living body has a DNA as a genetic material. Then in 1961, two scientists were there, Frank Jacob and Jacques Manon. They proved that process of protein synthesis is done with help of DNA in bacterial cells. They proposed a model of protein synthesis with help of DNA in bacterial cells. See, protein synthesis we will study in here, but protein synthesis is most important process in our body. Okay. thereby the technique of recombinant DNA. What the model, what is its advantage? Then advantage is that it helped to uncover the codes hidden in DNA. Now we will study about codes in these 3 points. In these 3 points we will study what are the codes. So recombinant DNA technology was increased. Okay, then in third paragraph you will see how the heredity is used, or in which useful, how the heredity, science of heredity is useful. The science of heredity is useful for diagnosis, treatment or prevention hereditary disorders, protection of hybrid varieties of animals and plants. So these are the uses of hereditary science. So here scientists we studied: George John Mendel, Godevirus, then Walter Sutton, Maclean McCarthy, Oswald Avery, Colin MacLeod, Francis Jacob Monod, Jack Monod. This scientist we studied. Okay. In further transcription, translation, and translocation. Before moving to these points, we should know some basic points. Those basic points are full form of DNA and full form of RNA. See, full form of DNA, deoxyribose nucleic acid. Full form of RNA, ribose nucleic acid. These are the full forms of DNA and RNA. Now nitrogenous bases in DNA, and nitrogenous bases in RNA. See, nitrogenous bases in DNA are adenine, thymine, guanine, and cytosine. Adenine pairs with thymine, guanine pairs with cytosine. That is of RNA is adenine, uracil, guanine, and cytosine. Adenine pairs with uracil and guanine pairs with cytosine. Here if you see the change in DNA, thymine is there, but in RNA, you will see U means uracil is there. This is the change. Okay, so these are the nitrogenous bases. If you studied the structure of DNA, see, if you see this, if you see this, on these lines, you will see the nitrogenous base. Okay, these are the points too you should know. And types of RNA, mRNA, rRNA, and tRNA. mRNA, messenger RNA, rRNA, ribosomal RNA, and tRNA, transfer RNA. Okay, now transcription, translation, and translocation. With help of RNA, the genes present in the form of DNA participate in functioning of cell, thereby control the structure and functioning of the body. So RNA participate with DNA. See, DNA and RNA. This both participate in functioning of control and structure and functioning of the body. DNA alone cannot do the functioning, or RNA alone cannot do the functioning, so they need both of them, they togetherly do this work. Information of protein synthesis. See, information of protein synthesis is stored in DNA, is stored in DNA. So this information of protein synthesis, information of protein synthesis is stored in DNA. Is stored in DNA means how, where, in which way, where is the requirement, in and what way the protein or and that can be synthesized. See, that is the important here. Synthesize of appropriate protein synthesized of appropriate protein through RNA as per the requirement is necessary for the body. Means which information is there in DNA, that information is essential to do the protein synthesis as per the requirement of our body. These proteins are synthesized by DNA through RNA. Again, see here, DNA through RNA means here DNA alone or RNA alone cannot do. They should togetherly do that. They do this work togetherly. This is called central dogma. One mark question: what is central dogma? Okay. This is called central dogma. mRNA is produced as per the sequence of nucleotides on DNA. See, mRNA is produced as per the sequence of nucleotides on DNA. Nucleotides, I told you the sequence of nucleotides. You know the nitrogenous bases key: adenine, thymine, guanine, cytosine. This is suppose the sequence ATGC. According to that, mRNA is produced. According to that, mRNA is produced. If here TACG, according to that, mRNA is produced. Okay. Only one strand of DNA is used in this process. This sequence of nucleotide in mRNA being produced is always complementary to the sequence in DNA. See, besides, there is a uracil in RNA instead of thymine in DNA. I told you the nitrogenous bases. Okay, in DNA, nitrogenous base, one nitrogenous base is thymine, but in RNA, one nitrogenous base RNA consists uracil, DNA consists thymine. So in DNA, thymine is founded, and in RNA, uracil is founded as a nitrogenous base. Okay. Now this process of RNA synthesis is called as transcription. So here you can see the picture. you can see the picture of transcription. If you see here neatly, you will get RNA polymers, RNA nucleotides. Okay, and here transcription is going on. So here if you see neatly, you will get the nucleotides, MAUC, sequence you will get. Okay, in this form you can go through. Okay, the mRNA formed in nucleus comes in cytoplasm. See, now what happens ki the mRNA which was formed in a nucleus, we have seen it was formed in a nucleus. Now it came to a cytoplasm. Now it came to the cytoplasm. It brings in the coded message from DNA. So what it brings, it means a coded message. Now what is the coded message? Now as you know key see, as you know, code, take an example. Code means we can say problem, coded message. As you know, all information about all information about the protein synthesis is stored in DNA. Is it correct? All information of protein synthesis is stored in DNA. Therefore we can say, therefore the mRNA brings the coded message from DNA. Why it brings the coded message from DNA? Because all the information of protein synthesis is stored in DNA. So DNA gives the coded message to mRNA, and the mRNA gives or comes with that coded message to the cytoplasm. Okay, now 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 the next we'll go through is the message contains the code for amino acids. See, this is the most important key. The message contains code the code for amino acid. Acid consists of 3 nucleotides. It is called a triplet codon. It is called as a triplet codon. See why this is called as a triplet codon? Why this is called as a triplet codon? Because one amino acid, take one amino acid need three nucleotides. One amino acid need 3 nucleotide to know what is the code. Due to this, the bunch of or the triad of 3 nucleotides is called as triplet codon. Okay, triplet codon. Then now here is a one scientist. You will see Dr. Hargovind Khorana, who was an Indian. He founded the triplet codons. He discovered the triplet codon for 20 amino acids. Okay, for his work he got a Nobel Prize in 1968. Okay, then each mRNA is made up of thousands of triplet codons. See, each mRNA is made up of thousands of triplet codon. As per the message of mRNA, amino acids are supplied by the tRNA. See, the message is with mRNA, and the amino acids means the message is problem, the amino acids will give the solutions. Means amino acid is finding a problem on that code. So that amino acids are brought by tRNA, has anticodon having complementary sequence to the codon on mRNA. Now what is the anticodon? Here we will go through. See, anticodon means see, this is the code. We said that code is a problem. And anticode, as you know, it is opposite. So problem opposite is solution. So anticode is a solution. Now this solution is given by the tRNA. This solution is given by the tRNA. Why? Because tRNA consists of amino acids. tRNA consists of amino acids. So the code is with tRNA. Okay, this is simple. Then 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 tRNA. Each anticodon having complementary sequence to the codon on mRNA. This is called translation. Now word translation is simple, isn't it? Word translation is simple. When you heard key translation means to translate something, you know, language translating English language to Marathi or Marathi to English. This is a simple translation of the language. Same way, same typely, or in the same manner, codes are translated to the anticodes. Problems are translated to the solutions. Okay, this is the translation. This is the translation which tRNA does. tRNA have anticodes, mRNA has codes, and they translate codes to the anticodes. Hence, the protein synthesis takes place. This is a very Now, amino acids brought by tRNA are bonded together by peptide bond. Amino acids brought together are bonded by the peptide bond. See, this is the important. With the help of rRNA. During this process, the ribosome keeps on moving from one end to mRNA to the other end by the distance of one triplet codon. This is called translocation. This is called translocation. Now see what is translocation. See when translocation. See, suppose this is the code. Here I will draw a sequence of nucleotides. As you know, first is I say A means adenine, then I say thymine, then again I say guanine, again I say cytosine. Okay, this is the we can say the code. Now this code, what I'm saying, this code is located means to know the problem. What is the problem? To that, the ribosome. Suppose this is the ribosome. See, this is the ribosome. This moves from one end to the other end with the distance of one triplet codon. That is nothing but translocation. That is to locate what is the problem, is nothing but translocation. It is very simple. Okay, now after translocation you will get after translocation you will get after translocation a most important problem we will go there, that is mutation. Now in first paragraph we studied that Hugo de Vries was a scientist who studied the mutational theory or the changes in the mutational theory. That scientist, on that basis this mutation is done. Now how the mutation is done, I will tell in a complete simple manner. See how the mutation is done. See, this, suppose that this is the DNA. Okay, first I will explain you what is it. See, when we know कि in 9th standard we seen कि genes are transmitted from parents to offspring. Same way genes, you know, genes, whatever it is, hereditary genes, so hereditary material is transmitted from one generation to the another generation. Okay, so that the genes may go correctly, neatly, very not unwrongly, but sometimes the sequence in the nucleotide of the genes changes. sometimes the sequence in the nucleotide of the genes changes, due to which mutation occurs, now if you know ki coronavirus, yeah, you know with coronavirus, now it is very it is following the great vast um, mm, vast place it has acquired. Same way, why this is the one coronavirus. Now in this one coronavirus mutation occurred, and these were the some characteristics. Suppose here four characteristics were there, that four characteristic have become five, or sometimes they become, instead of increasing they may decrease. This mutation occurs in a coronavirus. Same way, why this mutation occurs? Due to the change in the we can say change in the nucleotide sequence. Same way in humans, sometimes the see, this is a adenine, thymine, guanine, cytosine. Again I will say adenine, and again thymine. See, this is sequence, you can say. Now this is the correct sequence of the parent. Now this sequence offspring should get correct. Okay, instead of CCA, see, this is here CCA. Instead of CCA, here instead of CCA, if TAG, this sequence came, it creates a problem. Some mutation may not be, now some mutation, what we say, some mutation may not be very minor, they may be minor, but some mutation may be major. Due to major mutation, a most worst and vast hereditary disease, that is sickle cell anemia can be caused due to major mutation. Okay, this is the simple part, okay. See here also, same way they have shown, see here also in textbook, same way they have shown key the GAT. See, this is the main sequence, this is the main sequence, this is the correct sequence, you can say. Now here in this is the correct sequence. Here GAT was replaced, but the correct sequence is here middle, TGC must be there, but the GAT replaced by TGC. So this may be minor, what I'm saying, this may be uh, this may be minor, but sometimes this may not be minor, and the unminor or major problems can cause, here they have told key, the major problems can cause sickle cell anemia, which is the very 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 worst disease. Okay, this was about uh, these 3 points. We have studied key hereditary and hereditary changes, and then transcription, translation, translocation, and mutation. So these points we studied. Okay, mutation we studied. Further points we will study in my next session. Thank you for seeing this video. Please like, please subscribe, please share, and please press the bell icon, due to which you will get the notification of my new video. Okay, thank you friends. Okay, I'll stop here.